Dame Janet, to you.
Thank you very much indeed, and I hope this is going to work okay. So I was asked to give a talk that might appeal to this group of people, and I'm just not sure I've done that, but I will try my best and tell you about my world, my academic world, which is about understanding protein structures. So I'll broadly give a very brief introduction about proteins, from the smallest part to the very largest complexes. So we know that all living things are made from the same stuff, and that basically includes DNA and proteins. So whether it's human beings or wheat or fish, or you know that the first human genome was sequenced in 2001. It's basically 3 billion letters of DNA code. And for human beings, that codes for about 21,000 genes, and each of these genes makes a protein. So you will also know that the cost of sequencing was huge at the beginning of this century. It cost more than the cost of the most expensive house, or one of the most expensive houses in London. But today, the cost of sequencing a genome is only about $1,000. And so we get more and more information about genome sequencing. And the challenge is to try and interpret the sequences in terms of the biology that they do. And this is similar, this is just to show DNA provides the instruction, like when we're building a bookcase, you know this is the bookcase, but you get it all in bits. The DNA provides the instructions that comes when you buy a flat pack piece of furniture. The DNA is the flat pack instructions to create a human being. But the human being itself, although it contains DNA, is fundamentally made up of proteins. And of course, you're all familiar with proteins in the sense that you know that you should eat some proteins, and that comes in many shapes and sizes in all living things, in meat, in beans, in rice and nuts and fruit, of course, and milk. But proteins actually do many things. So what are they? They're made up of a polypeptide chain of amino acids here. So this is the DNA sequence that I talked about, GCC, TGC, etc. There's 3,000 million of those. But each 3 of these code for an amino acid. And these amino acids, so we have a long chain. Each gene corresponds to a chain of protein, each protein corresponds to a chain of amino acids. And some of the amino acids are seen here. And it's a little bit like you can think of it as a shoelace, a very long polymer, or you can think of the side chains as being almost lollipops on a stick. So you have the lollipops here and here and here and here. They're all different. Well, there are 20 of them, but they all are attached to the backbone, which goes along here, which is the polymer that joins it all together. And we know that this sequence of amino acids that's illustrated here, this long chain, and most proteins are about 300 amino acids long. So we've got 300 amino acids that then fold up into it. So they start off unfolded, but they then, of their own accord and spontaneously, fold up into a shape. And it's that shape that dictates biological function. So these structures that we see are determined using different methods, mostly up to now crystallography. And here you can see the synchrotron in Grenoble, where structures are determined. but there are now many synchrotrons in Europe. You can also use NMR, but recently electron microscopy has made it much easier. But actually, proteins are tiny. They're much too small to see by eye. In fact, so if you think of your credit card, that is about 8 million angstroms thick. A small protein like this one here is only about 50 angstroms in diameter. So you could fit lots of proteins into the sort of thickness of a credit card. And the distance between neighbouring carbon atoms is about 1.5 angstroms. So these are very, very small molecules. And until recently, until last century, 1960 odd, we didn't have a clue about what they looked like. And it was the pioneering work of Kendrew and Perutz in Cambridge, UK, where they determined the structure of the first protein, the myoglobin. And they do this by measuring electron density. So this is the blue mesh that you can see. And the blue mesh represents the electrons in the protein. The protein first has to be crystallized, and then you have to fit the atoms of the protein to this. Now on a computer screen, it used to be done in a big, something called a Richards box, but now it's all done computationally. So I was just saying that the first structures of proteins weren't determined until the 1960s. But since then, we've learnt a huge amount of protein structures. They're built into these electron densities, and you can see one here. And this should rotate, but unfortunately, it isn't doing so. But this is in 3 dimensions. It's very important to realize that this is a 3-dimensional object. And so you can represent, it looks in this representation as if there's lots of holes in it. That's not true. In fact, it's a solid object that looks like this. And we can ask, well, what do these things do? Well, in fact, in life, in anything that is live, proteins do almost everything. So they provide the structure in your skin, bone, and hair. They allow you to move. They perform the chemistry, the catalysis that's involved. They transport the oxygen around your body or iron. They provide all your immune defenses, the antibodies and the T cells. They're responsible for transmitting all your nerve impulses. They control expression, and they provide the messages between cells, the hormones that tell cells what each other's doing. And so proteins are absolutely fundamental to life, all life. And here you can see insulin, that you're well aware when you eat a meal. Insulin is secreted into your system so that you can promote the absorption of glucose from the blood into the fat, liver, and skeletal muscles. And again, I'm sorry, this should be rotating. Here we have collagen. That provides the connective tissues, the main component of gelatin, and it's critical. And you can see this is a sphere representation of these solid molecules. And this is Herceptin, which is an antibody that's used to treat breast cancer. And lastly, we have Botox, which is sometimes for some people they inject it, or more for cosmetics, I think. And this is, as you see, a surface representation. This looks like a solid blob, a ball, and this is the protein. So what I really wanted to talk about was how these proteins are chiral. They have an inherent chirality within them, and to understand where that chirality comes from. Now, we're aware that human beings, of course, are achiral. We know that we have our heart on one side, our liver on one side. Some things we have two of, so they're symmetric, but we certainly are mirror images. So we have an asymmetry arrangement inherent within ourselves. And this asymmetry, in a way, comes from the basic asymmetry of protein structures, part of it. And proteins are made up of these amino acids. So I should have, if I go back very quickly, oh gosh, this is too far to go back. Where are we? Right here. We have these amino acids are made up of a basic unit of chemistry. Each one has you can see we've got the red, green, blue, red, green, blue, red, green, blue, red, green, blue. So this unit around here, so we go green, blue, green, 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 blue, green, blue, green, green, etc. So when we look at the basic chemistry of these molecules, we see that the amino acids are actually chiral. So if we have our nitrogen, so that would be in this molecule, you can have the L amino acid or the R amino acid. And in fact, there is a hydrogen, so the R is the side group. And you can see these two representations are mirror images of each other with the plane of symmetry down the middle, if you like. So they're mirror images. And all proteins, more or less, there are one or two, but more or less in human beings, all proteins involve L amino acids without the D or the R amino acids. So it's critical that we have these, and this is right at the very heart of protein structures. It's each one of the amino acids has this inherent chirality within it. And you can see this from this picture. You can see in this case, you've got the mirror image here, and it all depends on whether the hydrogen is here and the side chain, the R group, is here, or vice versa. And because this is in 3 dimensions, and I hope you can get the 3-dimensional impact here, you have these 2 different possible types, but proteins only have this. And the hydrogen is very small, but the side chain, as you saw in that picture, there are some that are really quite large and involve up to 10 amino acids. So, 10 atoms. So the R group is big and the H group, the hydrogen, is very small. And that means that you have this asymmetry where one side has a bulky R group, the other side has the little hydrogen. So what does that do? So here you can see this is the chain of the protein coming through the center here. So the CNCC, CNC, or perhaps I'll do it like this, N C alpha C, N C alpha C, and so on. Now, these bonds that connect the atoms, the nitrogen, the carbons, and the oxygens, they're flexible. And in particular, these two, the connection between the nitrogen and the C alpha, here the phi, and between the C alpha and the C, the psi angle, those can rotate. So you get a possible rotation. And here, this C alpha, that is where you have the asymmetry. That's where you have the L amino acid. So if you like, coming towards you will be the hydrogen. Oh, no, it's the other way around. Coming towards you will be the R group, the big side chain. Going away from you, attached here, will be the small proton. And when you rotate these atoms here, you get clashes between this peptide group and this peptide group here. And so you can't just freely rotate this. So it's not like a shoelace where you can bend it in almost any direction. With this main chain, you can only rotate it in certain places because otherwise you get steric hindrance between the atoms. So this nitrogen will bump into this nitrogen. And so you have to be you're only allowed certain conformations, they're called, of this chain. And here you can see, I don't know if this is really where you need the rotations. You have the chain of the peptide plane here. And when you rotate about these bonds here, then you get clashes. So you get asymmetry. So the polypeptide chain, although it's a polymer, unlike many other polymers, like many plastics, it can't rotate freely about all of its angles. It actually can only adopt certain angles. And so Ramachandran in India in early 1963, so this was about the time that the very first protein structure was being determined in Cambridge. And Ramachandran actually built a physical model of this polypeptide chain here, a dipeptide, just 2 units basically he built. And he worked out for the phi and the psi, so that this is these two angles here, the phi and the psi, he worked out which positions were allowed and which positions weren't allowed. And he generated something which is now called the Ramachandran plot. And in this plot, he said certain regions are allowed and other regions in the middle here are disallowed by the chain. So this plot, which now is drawn like this, actually represents the allowed regions of the Ramachandran plot and the disallowed region. Now, if you had, this is for an L-amino acid. So these are the allowed regions, and these are this is the equivalent for a D-amino acid. So you can see straight away that having L-amino acids constrains the structure at its very basic level to certain parts of this Ramachandran plot. Had we had D-amino acids, then it would look like this, and we would end up with the opposite mirror image. So the consequences of having this Ramachandran plot is that the polypeptide chain tends to adopt the alpha R conformation here, the alpha helix, and the beta strand conformation here. And there's a small area here, the alpha left conformation, that is mainly adopted by glycine, which is a residue which doesn't have a side chain, so it is achiral. So if we look today at all our structures and look at where their phi-psi angles occur, so this is the phi and the psi, we see that almost all of them are red, in these red regions where they're allowed to be. And so this then generates for our polypeptide chain a certain chirality. And that chirality determines that we have alpha helices that are right-handed. If we have the D-amino acid, we would have alpha helices that are left-handed. So if we look at the polypeptide chain, it turns out that it will, of its own accord, because it's a very stable conformation, wrap into a right-handed alpha helix. Because of the chirality of that amino acid, it will never, almost never, adopt a left-handed helix. It does very occasionally, but generally it doesn't. So if we look at that, then we are adopting, this is the, if we have phi-psi angles here in this big red region, then we adopt the right-handed alpha helix. And that is an implicit part of almost all protein structures. The alternative region of the Ramachandran plot is this big red region up here, the beta region. That region creates not helices, but strands. And I don't know if you can see this because it's really not very clear. You have the strands in a sheet, and actually it gradually rotates because of the chirality of the alpha carbon. It will rotate round. And you can see here, this is again rotating round. So we have not flat sheets, but actually twisted beta strands as well. So the consequence of that is that if you have here, we have a chain that is going round like this, but you don't just have a hairpin which is planar. Instead, it twists round as it goes. And you get that twist is always in the same direction. And so when you have, hang on, I'm going to go right back, actually, here. I think you can see in here, the chain is folding into these into these helices. Can you see the helices? They look like helices there. That one's different, but here you can see the strands. Can you see the red strands at the top in the herceptin molecule? And you can see that they are twisted. They're not flat sheets. It's like getting a piece of paper, getting hold of both ends and twisting. And all sheets are twisted, and they're all twisted in exactly the same direction. So that small change in the chirality of the basic amino acid results in twists in these beta sheets, and the helices that are always right-handed. If you look at these helices, you can see one here. It twists round and round, but always in this right-handed direction. And so So we have these very clear chirality at the secondary structure level. But in fact, the chirality goes even farther, because you can then think about joining up. When you have a long polypeptide chain, then it will create 3 helices on a trot, or 2 strands on a trot. And you can see here, you get this twist, which is left-handed, and here you have a twist, which is right-handed, and the same for this connectivity here. And so these are called motifs. In the case, usually, actually it's not. So we can have then beta alpha beta, or a beta alpha beta, and they can be connected. So if you start here, you go around here, come to here, that's a left-handed connection. And when you start here, this is a right-handed connection. Now, in fact, when I was a postdoc, my very first postdoc position, the first, or one of the first projects that we did was to look at the handedness of these beta alpha beta motifs. And we were amazed. I should say, in those days, we couldn't really do it on the computer. We used to use stereo glasses and look at these pictures in the publications to see whether these units of the structures were right-handed or left-handed. And we were amazed when we found out that almost all of these units, about over 90% of the connectivity between these motifs was right-handed. And in fact, this was for depending on what you got. Sorry, that's not very good numbers. I'll go past. So essentially here you can see we have a very simple beta alpha beta unit, one taken from actually alcohol dehydrogenase, and you can see it's beautifully that conformation. Now, if you look at it, you'll see that these sheets as I said before, they're each twisted, and when they form into a sheet, the whole sheet twists. And by forming the right-handed connection, you can see that that connection is much more efficient than actually going round in the opposite direction to connect in the opposite left-handed way. And so when we were doing this, and I did this with my colleague Mike Sternberg, finding that over 90% of these connections were right-handed was truly amazing for us, because almost every other rule in protein structure is only obeyed to about 70%. But this particular rule is really powerful, and it comes from that chirality of the individual amino acid that determines the twists in the sheets and then the best way to connect those strands. Here you can see a very complicated parallel beta strand motif. And here you're coming along here, up round all this complexity and back in. And that can be measured as a right-handed beta alpha beta. Now, I had a student in about the early 90s who wrote several algorithms to determine these chiralities automatically from the coordinates of the protein. So we have our handedness of the beta strands and the handedness of the beta sheets. And again, this connection, which is it's sort of ugly. This is rather beautiful and simple, whereas this connectivity is much less appealing and is not really seen. So when we look for the beta strand, where we have, as I say, about 92% here we found were parallel beta strands, and a very small amount were complex. Most of them were complex, and a very few were left-handed. And this, so this is we've talked about the individual amino acid, we've talked about the secondary structures, we've talked about the so-called super secondary structures, and this is another one. This is a Greek key motif. And again, in this case, you have this connectivity from A to B to C to D. And that is fundamentally a right-handed Greek key. The opposite, A, B, C, and D, again, is not found in proteins. So it goes not just for one connection, but for building up these motifs that form the core of these beta streams. So we actually have more and more and more structures. So it became quite interesting to use these structures, try and understand how they were related together, and to look at the complete fold, or as we call it, the topology of these proteins. And you can see many of them have alpha helices. You can see that they have the arrows, the beta sheets. Here's one that's nearly all beta sheet. And you can see that they sometimes have repeats, and there's internal symmetry within them. And here you can see one that's just alpha helices. But when we saw these, we wanted to try and classify them. You'll be familiar with botany and aware that originally Fuchs and other people classified plants according to how similar they looked. So they asked what the leaves were like, they asked what the roots, what the shape of the plant was, whether it had flowers or seeds, all those things. And in a similar way, we began looking at all these protein folds and tried to classify them. And what we came up with, and this was with my colleague Christine Orengo, was a hierarchical classification of protein structure, and it was built on class, architecture, topology, and homologous superfamily. So it's a hierarchical classification, and we gave numbers to all of these to make it computationally similar. And when you do that, what we found at that time was that About a quarter of the proteins had just alpha helices, about a quarter had just beta sheets, and about a half of them were alpha betas in various different architectures. So you can see here, you sort of have a beta sheet in the middle surrounded by alpha helices on the outside. That's called a tin barrel, and I'll come back to that. Here you see you have an up-down barrel. Now that barrel actually has a handedness, and it has a handedness because the connectivity between the alpha, alpha, alpha is right-handed and not left-handed. And so we classified these domains there, which are like the Lego bricks of proteins. We found some folds were very common, and here you can see at that time these were the most common folds. And what we found was that also, and other people observed this as well, that during evolution, structure was much better conserved than sequence. So in fact, the globin that we have is similar to the globin that plants have, that whales have, that all creatures have. It's conserved over millions of years of evolution. And of course, these are complicated structures. You can see in 3 dimensions. Unfortunately, they're not rotating, but you can see they are very complex structures. And at one stage, we were trying to simplify them because you can see here the architecture is that you have a beta sheet in the middle where the strands are represented by triangles and the alpha helices on either side. So this was called an alpha beta alpha sandwich, a layered sandwich. And the connection, if you look carefully at this topology diagram that's generated automatically, there's a connection. So the strand is going up, it goes down into the top of the helix, out from the bottom, into the bottom of that strand and up towards us to the next helix. And this is a fairly typical alpha beta protein. So here, of course, we're not just involved in fact, when we make these Lego domains, these proteins often have multiple domains in, especially the eukaryotes, but they also form multimers. So they form tetramers, where there are 4 of them, or octamers, or many more than that. And so when we look at the hemoglobin, here you've got a stick representation. You can see the red blobs are the heme molecules that carry the iron in the red blood cells, but they have a symmetry to the way that they form these multimers. And again, this can be traced back to the L-amino acid through the secondary, super secondary structure to the tertiary structure and beyond to the quaternary structure. And this tin barrel, in fact, this I think is the most common fold that you see in nature. This has got the strands in the middle and the helices on the outside. And in fact, this fold, I'm not sure where the beginning of this is, you can't see the end terminus, but it always proceeds in the same direction around the barrel. So even the tertiary structure of the molecule is very much driven by this chirality. And here you have the quaternary structure. We've got the tobacco mosaic virus. This is, you've been hearing all about viruses, but in fact the TMV virus is a right-handed assembly of RNA in the middle and the proteins on the outside. And just like when you go in the garden and you see your sweet pea that always curls in the same direction, with the TMV, even at these tiny, tiny molecules have this inherent chirality to them. And it all comes about because of the chirality of the amino. So I think that's really where I would like to end. I hope that you've understood. It's really quite difficult to explain. I'm sure as mathematicians you can calculate chirality, and that's what we tried to do a little bit, but not very much. But it just still amazes me how you have these chiral features from the very tiniest sort of 4-atom core that then propagate out to the structure, the whole structure of the protein, and ultimately, in some senses, to the whole structure of the organism. So I think I'll finish there, and I should thank. I'm sorry, I've not got an acknowledgement slide, I don't think. But if I could just acknowledge. Oh no, I'm not. Hang on. No, I haven't got an acknowledgement slide. So I'll just acknowledge, in particular, Roman Laskovsky, who helped with some of these slides, and also Tim Slidell, who was the person who worked on some of this chirality for his thesis. And some of these old slides that you can see are black and white, are taken from Tim's thesis that was, I think, in 1996. So it's many years ago now. So shall I stop sharing now?
Okay, thank you. Well, thank you very much, Janet. A very interesting tour of information. I wonder if we have questions for you from the audience.
So it was really fun to see this overview. I haven't really thought about, I'm a DNA person, so I don't really think much about proteins anymore. Yeah. But, I'm always interested in what didn't happen. Is there a favorite thing that you said should have evolved, like a certain fold that should have evolved that we haven't seen yet that surprises you?
I think I've learned never to, almost never to predict, actually. So when the 4 helix bundle, we had, so that's just 4 helices on the trot, and it's just up, down, up, down. So it's a very simple bundle of sticks, basically. And all the examples were right-handed. And somebody contacted me and said, Janet, we've solved the structure of actually ferritin, which carries iron around, And he said, from the map, from our electron density, it looks left-handed. Do you think it could be left-handed? And happily, I said, well, so far they've all been right-handed, but you never know. You never know what's going to happen in biology. So I think most of the obvious folds have been seen, frankly, and you get they're just not as obviously in protein design now. People are making very, very, very regular structures, which are very, very stable. But this right-handedness, so I don't know if you know David Baker, but this problem, you get to the end of a strand. Do you turn right or do you turn left? And this is a real, and actually you really don't want to turn left because that will screw up the whole protein. So these inherent preferences are really critical, and they're built into all the because everything we see have these preferences in them. So if you're machine learning or whatever, you automatically pick them up from the structures. They're there. So I can't say there's been one, I've been wrong on lots of things, but I can't say there's been one that actually, well, no, there was one, I thought we should see solenoids. So you get them going round, and you could do it with alpha helices, like a Toblerone bar. And in fact, they showed up. So, so we have these strands, we have these like Toblerones with the strands going along one side, down the other, and round and going round like that. And we have them with alpha helices as well, solenoids, where they go round. So those kind of showed up. So but they are
Thank you.
amazing. Yeah.
Other questions? Janet, I have a question for you that is extremely basic, and I'm sorry for this. I'm just curious. I don't know if it makes sense to the question itself, really, but I was curious to understand or to know the twist that you talked about has more to do with the function or with the packing, a mechanical packing, assuming that, of course, mechanical packing also has its own reason. But I was just wondering.
No, the Ramachandran plot is basically the consequence of physics and chemistry.
Due to the atomic distribution along strands.
It's atomic, it's steric hindrance and electrostatic interactions. I've not talked anything about the sort of physics side, but the hydrogen bonds between the bits of the amino acids are absolutely critical. But they're not steric per se. It's this chirality comes totally from these basic L-amino acids, and it propagates right up to large complexes, which I still find, even though I worked on it many years ago, I still find it fascinating that that's the case.
Thank you. Sure. Questions?
I have a question. Can everyone hear me?
Yes. Eva, you're talking.
Hi. So I'm sorry. It's actually Sophie Jackson, not Ava.
Sorry.
Hello, Janet.
Sorry, Sophie.
It's a lovely time. Sorry, we're using her laptop because, as usual, I forgot mine. I'm just. It was a lovely introduction, Janet. So again, with these kind of super secondary motifs, where there's a distinct preference in nature for one-handedness over the other, I couldn't quite tell. Are there any entropic considerations? For example, the loop has to be longer with one-handedness than another-handedness. So is it an entropic effect as well?
I think so. The loops, there are some left-handed connections, and when there are, they are longer. There's been some work that suggests that these left-handed things are often associated with function. I'm not sure statistically that's been validated. I personally haven't looked at that recently. I think there's no doubt, if you look at the Ramachandran plot, that is effectively the population plot, but it translates into an energy plot. And on any basis, an amino acid in a loop, let alone in a, will spend more of its time in right-handed space than in left-handed space, if you see what I mean. Yeah. So there is an entropy effect there, I think. How much I think it just doesn't fall. I mean, it still amazes me, frankly, how strong this preference is to go for right-handedness in these things. So I think it's just to do with the packing of the helix. It nestles rather nicely. If you have a beta alpha beta unit and you pack it, it kind of nestles in between not in between them, but on top of them quite nicely. Whereas if you have to go the long way round, it's all a bit clunky. And there are kind of holes. It doesn't naturally sit quite, but that's a lot of hand waving. I've not done the entropy calculations.
Okay, thank you.
Yes, other questions? No? Okay, if not, let me thank you, Janet, again. Thank you for the wonderful talk. Thank you very much.